Shannon is an AgResearch senior scientist in the Animal Genomics group based over at Invermay. She has a molecular biology and biochemistry background both in plant and animals. Her research area presently focuses on genomic tools, development, implementation in not only livestock industry but in agriculture industry. I'll just hand over to welcome Shannon.
So I'm going to talk about the sheep genomics project, so basically about generating all the genetic data that's going to feed into all these pipelines that we've heard today. So the project overview is to generate genetic data that feeds into this genomic selection and gene discovery studies. This is going to come from whole genome sequencing. We've heard John allude to the fact that it's a lot cheaper now, so $1,000 for a human genome, and sort of animal genome sequencing is just a little bit behind that. It's sort of between $2,000 to $3,000 per genome. We're also going to use a combination of the 5 to 15K low density SNP chips and also this high density SNP chip that's been recently developed by the International Sheep Genomics Consortium and PharmIQ. We're focusing on maternal and on-farm traits, and the idea is to have greater genomic discovery. This is for causative mutations, genes and genetic pathways. And having these greater genomic discovery, we're hoping to have a better understanding of the actual biology of the trait, higher accuracy of prediction, so more accurate GBVs, and better persistence of accuracy across the generations. Results from year one, we've been sort of focusing on the generation of additional genomic data. This is through high-density genotyping of the 2013 industry sires, and Anna has been busy out collecting for the 2014 industry sires. We've also been updating our historic data set. So within the Avita program, there's about 20,000 sheep, including over 13,000 sires, have each been genotyped with the ovine 50K SNP chip. Now we've updated this data set by imputation to the high density of 600,000 SNPs. A very simplistic look at imputation. So on the top line there we have a representation of, say, whole genome sequence from an animal. In the middle is high density genotyping and then 50K genotyping. So if you have key animals that have been genotyped, say, with a high-density genotyping and others with a 50K, you can basically impute or fill in the missing genotypes from the relationship with the high-density genotyping. And then with the more whole genome sequencing we're hoping to do, we'll be able to do the same thing, where we can fill in all our HD genotypes with the whole genome sequencing. So just a sort of a schematic of this, we have a whole lot of animals that are genotyped with a 50K SNP chip, and we're basically going to fill in the rest, so we'll have effectively the 600K for these animals now. This imputation relies on a reference sense of animals being genotyped with a high density SNP chip, so we've got about 2,000 of these that have been used, and they're predominantly the key industry sires. And just a representation of this, so the ones in red there have been genotyped with the HD SNP chip, and you can see it was representative of the others that have been imputed to the HD SNP chip in black. Just to let you know, that's the breeds basically. So Romneys are in the blue, Cooperworth are in the green, Texel yellow, Perindales are purple. So from the gene discovery parts, we've taken all this imputed data, we've combined it with all the phenotypic data from standard cell traits, and we've just performed some preliminary genome-wide association studies. This has been the work done of Suzanne Rowe. We've looked at 8 traits just to begin with: weaning weight, live weight, eye muscle area, fat depth, fleece weight, fibre diameter, GGT testing, and fecal egg count. And just an example of what you do get from these. So if each one of these graphs is for a particular trait of interest, along the bottom we have the number of chromosomes in your genome. Each spot basically represents a SNP marker, and up the y-axis you've got your significance. So basically where the red dotted line is sort of where the level of significance, and any sort of SNP markers or dots above that, they are likely to be SNPs that are associated with that trait we're looking at. So just to highlight with this one, so for GGT, where the green and the blue are SNP markers that are from the 50K dataset, and the red and the black circles are from the imputed HD dataset. So you can see with that high peak there on chromosome 15 that it's a lot more powerful using the high density SNP panel. So this coming year we're continuing to do genome-wide association studies in the hope of actually identifying the causative SNPs. And this will be then inclusion on low density genotyping, whether it's low SNP chips or genotyping by sequencing. We'll be doing the HD genotyping of the industry size. Now, we're also commencing the whole genome sequencing of about 100 influential sires. And this is going to be part of a 1,000 genomes project, similar to the Bull 1,000 genomes. So, but basically, by doing this sequencing, we'll have access to another 900 from around the world. And the aim is to maintain the genomic resources and to help ensure the New Zealand sheep industry continues to have access to the latest and relevant technologies in genomics. Thanks.
How much more accurate is the high density than the 5Ks? 5Ks are
So currently with the way we're going to use it for more accuracy is actually being able to identify the actual causative mutations. If you have the causative mutations within your genomic selection, then those mutations aren't going to, then it's not going, those SNP markers are not going to change over the generations. So that's where the accuracy will increase, as opposed to a lot of them now are current have a marker associated with the trait, not actually the causative mutations.